Okay, well, I think since it looks like everybody is here and has been incredibly punctual, which maybe that's a reflection on Tim, because Tim tends to be very punctual, I would say. So, as just an introduction, so Tim is a native from Minnesota, and he went to Carthage College in Wisconsin, where he majored in chemistry and entrepreneurial studies. And I really didn't know how that was going to play into his PhD here in Bozeman when he first started in the group, but as you'll see, he somehow worked that in. And he has an ability to work a lot of different things into what he does. So in addition, so today what he's going to present is really probably less than half of the work that he's done. But it's a nice story that really kind of, he's put together and really done a nice job with this protein sensor assay. But he's been involved with a lot of other projects. He's involved with a DoD project we have looking at biofilms and trying to resensitize biofilms to antibiotics for treatment. He's been involved in a number of projects that haven't worked. And that's one of the amazing things about Tim is his enthusiasm. I don't know how many times we've gotten through months of work, and then all of a sudden we conclude, well, we're going to have to repeat it. Well, instead of getting depressed, Tim just smiles and takes this as an opportunity to make it better. And he really has, I think, ingrained this pursuit of excellence, which I think that's really going to suit you well in the future. So in addition to the science, he's also the guy who builds the computers in our group. Our IT specialist. He is our 3D printer expert, which I think many of you have seen the prints that he makes, which some are fun, yes, but most actually are highly useful and really help us in the group. So let's see, is there anything else I wanted to say? So he actually has a postdoc lined up, so he's accepted a position at Johns Hopkins University with Raoul Dingelsman, and he'll be studying malaria. And this is a really nice opportunity where he's going into a group that is primarily a proteomics group. And so he's going to get the opportunity to really learn proteomics, and he's going to take with him his expertise of metabolomics, which is what this group wants to get into. So I think it's a really nice position for him to go into, and I know that his future mentor, Raoul, is really excited for Tim to get there. He can't wait for Tim to get there. So, yeah. So, with that, Tim, take it away.
All right. Let me just make sure I get mic'd up here. Our cinematographer wanted to record me, so. Thank you, Brian, for that introduction. Thank you, everyone, for coming here this afternoon on a Monday afternoon. I'm sure you have better things to do than come listen to my research, but it means a lot that you came anyways. I'm really excited to present this research because, as Brian said, it's been one of the things that I've worked on for the longest, about 4 years now. And kind of unlike a lot of the other metabolomics projects that were going on in the group at the time, and I thought I was going to be a part of, this idea was born just kind of literally out of an idea Brian had from a past presenter. And I'll still remember the day Brian came into lab, and he told me he had an idea for my thesis work. And I said, okay, great. Thinking again, I was going to be working on some sort of metabolomics project involving cells or an organism, something fairly specific. And the first thing that he asked me is, do you remember a guy, Eric Anslin, who came to give a talk here at MSU a couple of months ago? And I said, I don't really remember Eric, so can you refresh my memory? So Brian told me about Eric's work. What Eric was doing is using the protein bovine serum albumin. And he was basically binding a dye, a reporter dye, inside of this protein's pocket, mixing it with perfumes or olive oils, and he could differentially sense, so to speak. He could tell the difference between one perfume and another, one bottle of olive oil or another, using this differential sensor in that of bovine serum albumin. But there was a problem. he couldn't tell which fatty acids or which terpene molecules in perfume were binding. So all he knew was that he could tell the difference. Brian thought we could improve upon this by adding a mass spec aspect to it. Brian does a lot of mass spec in his lab, and he said, well, let's see if we can apply this same kind of methodology, but incorporating mass spec. So why did we actually choose bovine serum albumin or serum albumin in general? Why did Eric work with it? It has to do with the properties of the protein itself. It's a protein found in the plasma of anything that has blood, really. And it was initially, we think, developed to transport fatty acids, potentially metal ions like copper through the bloodstream, which is highly saline, and those things don't like to go into saline solution, so it would transport them to where they needed to be. More recently, it's been shown, as you can see in this human serum albumin picture, there are drugs. In fact, we think most of your drugs actually bind to serum albumin. It's a huge thing that pharmaceutical companies study, is the binding of their drug to serum albumin. So we wanted to take advantage of that. Furthermore, each of these pockets that serum albumin possesses has different affinities for different types of compounds, so it can bind things selectively, but it acts kind of like a sponge. And we thought this would be a really good protein to use as a differential sensor, again hooked up to a mass spectrometer. A number of people have come up to me and said, well, what's that stringy spaghetti thing located up on the board upstairs? So this is actually a 3D model that I printed of bovine serum albumin. I forgot to bring it down here in my rush, unfortunately. But essentially, it looks very similar to the human varietal. It's a heart-shaped protein with 6 pockets to bind. Okay, so as happens with most PhD research, I think, you spend a lot of your time working on methods or collecting data, and one slide can summarize it. So 8 months of work trying to develop this assay I can summarize in one slide. What we call the protein sensor assay has 3 distinct steps. You have binding of molecules to bovine serum albumin from a solution. In this case, I'm indicating them as these little small molecules or shapes. Some of those molecules will stay bound to the protein, while others we can wash away. Those that actually do stay bound, we can then elute by denaturing the protein, and we can analyze those by mass spectrometry and generate a profile or a fingerprint of what was bound to the protein. And this is what we explored. Again, this took about 8 months, and I just summarized it, so yay me. Okay. First, I want to talk about what a mass spectrometer is. I'm not sure everyone's familiar with the basics behind mass spectrometry. So mass spectrometers kind of all function in the same way. They have some sort of inlet. In this case, I'm going to be using liquid chromatography, so I can separate molecules based on polarity before they enter. This decreases the number at any given time that are being measured by the mass spectrometer. A mass spectrometer itself is just a giant balance, okay? It's very, very good at measuring the weight of a molecule, and it does it very quickly. We can measure thousands of molecules in a rapid amount of time. In order to do that, we have to generate ions, so we have a source. In this case, I'm using an ESI, or electrospray ionization. It creates charged droplets, which then transfer that charge onto the small molecules that we can manipulate inside the mass spectrometer. For analyzing those molecules, we use what's called a QTOF, or a quadruple time of flight. Basically, all this is we have a giant tube, and when you push molecules into that, things that are smaller in mass will travel faster through that tube than larger things, so we can separate them based on their essentially mass. They hit the detector, and we get some sort of output. This is called an ESI QTOF mass spectrometer. It's the instrument that I actually used. In fact, here it is, the instrument down in the facility. On the left, I'm showing our Agilent 1290 liquid chromatography system. So again, inside of here is a whole bunch of plumbing, and this allows us to separate things based on polarity, in this case, of the molecules. On the right, you're seeing the Agilent 6538 QTOF. This is the instrument. This is my baby. I've worked on it for 4 years. Here you can see the giant flight tube. In fact, I can't even keep it in the picture. So we send molecules in there, and they'll get separated based on their size. Shown right here, inside of that white cover, and also right here, that's the electrospray ionization source. So inside there, we're creating our ions that we can manipulate. And this giant box contains a whole bunch of optics and lenses that we use to manipulate and push the ions so that we can actually detect them. Now, this system is fantastic. I said we can detect thousands of molecules in a very short period of time. That comes with a problem. You're detecting thousands of molecules in a very short period of time. A human brain can't analyze thousands of things very quickly. So we like to use something that'll reduce that down to a quick and easy picture that a human can understand. And the most common way to do that in metabolomics, the research that I'm studying, and in fact many omics, is to use something called principal component analysis, or PCA. PCA is a way to basically take your data from a whole bunch of metabolites and condense it down to a single data point for each sample. Now, I want to walk through a basic idea of how this works so that you can understand when I'm showing these pictures kind of the background behind that. Imagine I had two samples that I analyzed by liquid chromatography mass spec, LCMS. Sample 1 in green, sample 2 in blue. And metabolites in these red dots are indicated for the number of times I've seen them in sample 1, further along this axis, along sample 2 here. So this dot was seen maybe once by sample 1 and twice in sample 2. And so a lot of times this is what we end up with, is a distribution of metabolites with varying intensities, we call them, from the mass spec between two samples. Now the problem is that this means nothing to me. This is a bunch of data points. I don't know if sample 1 is the same as sample 2. Maybe it's a little bit different. This is where PCA comes in. What we do is we take and we add a new axis to this data point along the point of most variation between two data points. Okay? You can kind of see that. Maybe this data point and this data point. We've added an axis. We can then take and add a second axis orthogonal to this along the second most variation. Okay? If we rotate this so that it's a little easier to see, now it looks like we have the data in a new set of axes. And indeed, that's the first step of principal components analysis. We take our data and transform it into a new set of coordinates. We call these coordinates principal components. Excuse me. There we go. First principal component lies along the most variation in the data. The second principal component along the second most variation in the data. And you can do this for as many data points as you have. You have that many principal components. Of course, we can't visualize past 3. Now, this is, again, only the first half. This still does not make a lot of sense to human minds. So what the algorithm will do is it goes in and scores each of the metabolites for how much they contribute to a given principal component or a given axis. So here, these blue circled metabolites and green circled metabolites contribute the most to the variation in principal component 1. We apply a scoring factor to that, and essentially we can say that these are the most important to differentiating those metabolites along this axis. Then we can condense all of these data points, okay, through this scoring algorithm that it does in the background, condense it down to 2 data points, where sample 1 is now clearly separated from sample 2. And you couldn't have gotten that from the raw data that I displayed previously. That is the power of this technique. Finally, I'll add that this is unsupervised. It's unbiased. I did not tell the algorithm that sample 1 and sample 2 are different samples. Okay? It did that on its own. It's a very powerful technique because it is unbiased. Okay? So, having described how I'm gonna, this is the vast majority of what I'm going to display. Let's get into some data. One of the first projects that I worked on is in an analysis of wine, and this is why I loved working on this project. Is Brian allowed me to work on wine and a number of other different alcoholic beverages for my PhD. Not many people can say that. Now, why do we get into wine? There is a reason for this. First off, it's a complex mixture of thousands of metabolites. And to someone like myself, I very much enjoy drinking wine. But if you bring me 2 bottles of wine, I probably won't tell you anything other than, yep, that's wine. Okay? And so for us, as scientists, what if we could take our newly developed protein sensor assay and apply some sort of metric that will tell you this bottle of wine and this bottle of wine taste very similar, even though they come from different vineyards or different varietals of grape? That would be awesome, right? Or that this bottle is different from this bottle, even though they're from the same grape varietal. You may not like one of them. That was what we were hoping to do initially. We also thought, since we're delving into what's inside of there, maybe we can validate something like origin or grape varietal inside of that bottle of wine. Maybe we can say, yes, this really is a Cabernet Sauvignon and it's not mislabeled. Or maybe there's a little bit of characteristic from something else that's not on that label. Maybe we can look into contaminants that are inside of your wine that you didn't even know about. And finally, I thought it would be interesting to maybe shed light on the effect that breathing has on wine. If you talk with loves wine and considers themselves a connoisseur, they'll tell you that you want to let your wine breathe, right? But how long do you let it breathe? Does each wine need to breathe for the same amount? Again, for me, I open the bottle and I drink it. So maybe I shouldn't do that. What if we can shed some light on that, again, applying an analytic metric? So to do that, I designed an experiment to test, first off, the breathing. So I took a bottle of wine, or actually a couple bottles of wine, and I uncorked them, and I analyzed that using the protein sensor assay. I then let the wine breathe for 30 minutes, and then took a sample for that to treat. And finally, I waited 24 hours, thinking that after 24 hours of letting it breathe, something has to have changed, okay? This has to be different than the other wine varietals. I started off with just 3 wines, a Cabernet Sauvignon, a Merlot, and a Pinot Noir. Shown on the top is what we call a total ion chromatogram. I'm going to show several of these, so let me just walk you through it. On the y-axis here is the total number of ions, or molecules, small molecules, seen by the mass spectrometer at any given point. Okay? So along here you can see that we have increasing molecules, decreasing, increasing molecules hitting our detector. Okay? Along the x-axis is what we call retention time. Okay? So how long does a molecule spend interacting with the column before it becomes loosened and goes to the mass spectrometer? So I'm going to show a lot of these. Now, just based on this total ion chromatogram, you can see that there's differences between the colored traces, right? There are peaks that are more intense in, say, the green trace than in the other traces. So initially I thought this is going to work. Okay, we found a way to tell the difference between bottles of wine. And indeed, at 30 minutes, you can tell the difference between the 3 varietals, okay? They're very far from one another. Remember that we're trying to separate or show that things are similar to one another. In this case, they're clearly different. At 24 hours, the wines are also different from one another. So this would indicate that even after 24 hours, the wines are still differentiable. I thought, well, maybe I won't be able to tell the time points apart. But in fact, I actually can tell time points of breathing apart in individual wines. So on the right, I'm showing both the 24-hour and 30-minute time points for all 3 wines. You'll notice that there's still division, kind of you can see here, of the 3 different bottles of wine, but in addition we can see that the triangles, which is representative of 24 hours, are separated from squares of the same bottle. So I showed that we can indeed separate time points and varietals. But this was a very simple 3 wine set. So I moved into a larger set of Bordeaux-style wines. Okay, we chose these because the Bordeaux grapes are considered to be kind of the best grapes for making wine out of. And we had help from a master sommelier over at the wine gallery picking these wines out. He selected for us a series of 5 pure varietal of grapes, and he selected 2 blended bottles which contain bits and pieces of varietals from each of the 5 pure varietals. And so I'm just showing a picture. I again applied the same breathing experiment to each of these 7 bottles. I let them breathe for, well, I didn't let them breathe. I immediately analyzed them after uncorking. I let them breathe for a half hour and finally for 24 hours. These plots indicate each of the individual bottles of wine across their breathing time points. And I'm not going to spend too much time here. Suffice to say that we can tell the difference, for instance, in this Malbec, the 30-minute time point and the 0-minute time point are differentiable from the 24 hours. And this was kind of the case in all 7 bottles. We could tell the difference between the time points. The one that's a little bit unique is this blend 2, as it's labeled. The 0 and 30 minutes are actually right on top of each other. Now this could indicate one of 2 things. Either this bottle does not change significantly after 30 minutes of breathing, and you could open it and immediately begin drinking it. Or conversely, it could mean that you need longer than 30 minutes before it reaches its peak drinking flavor profile. So this was informative, but I wanted to see could we still discern the wine bottles from one another at each of the time points. So that's what I did. At 0 minutes, 30 minutes, and 24 hours after I'd uncorked those bottles, the wines are all discernible from one another. Okay? If we kind of zoom in on the 30-minute, the one that I've made a little bit bigger here, I noticed something that was actually really interesting and kind of enlightening for this project. So if we look at where Red Blend 1, which again contains pieces from each of the pure varietals, it fell right in the middle. So here's a peer, here's a peer, here's a peer, peer, and peer, and it falls right in the middle. It's exactly where we would expect it to be if the assay is performing like it should. Similarly, the blended bottle number 2, which actually contains a slightly different set of combination, falls closer to those that it's actually nearby. You can tell that it's very close to this Cabernet Franc and this Malbec, which are kind of heavy as its composition. So we can still tell the difference between varietals at each time point. We can tell the time points at any given bottle from one another. Then what I did is something crazy. I put everything onto the plot all at once. And I didn't know what I was going to get out of it. But I actually got some meaningful insight into the PSA's application to wine. If we look at, for instance, these orange dots on the left, this is a Cabernet Sauvignon, okay? Those are the 3 different time points that we allowed it to breathe, which I've indicated over here. So you can see that at 0 minutes, it's actually quite far removed from the other bottles of wine. And as it breathes, it becomes more similar, it would appear, to the other bottles of wine. If we look at instead this Petit Verdot, you can see that, again, we get nice clustering of those individual time points, but the 0 and 30 minutes are almost on top of each other, very much similar to the other bottles of wine, and the 24 hours is actually very far removed. So this could be, again, shedding more light on this idea that different wines require different amounts of breathing, and in some cases, if you let it breathe too long, potentially you've now got something that you may not like to drink. Okay, so red wine works. I can do it with larger sets, smaller sets. The breathing effects I can track with red wine. But I wanted to check and see if it would work with white wine. I didn't have any reason to believe it wouldn't, but a good scientist always double checks. And what you'll see here is I had 6 bottles of white wine. I had 2 bottles of Sauvignon Blanc from 2 different vineyards. And I had 2 bottles of Chardonnay from 2 different vineyards. And then I had 2 blended bottles. And if we look again at the total ion chromatogram, we can see that there's clearly differences just based on the chromatography from the LC data, the raw data. But I used another technique called hierarchical clustering, where the vertical line indicates the amount of dissimilarity between samples. And you can see that individual bottles, replicates of the PSA analysis on a bottle are very, very small vertical lines, meaning that the replicates are all very similar, whereby the other line, the ones that I've indicated and left in blue, are very different from one another. And this would denote that, for instance, these Sauvignon Blancs actually have some subtle differences between themselves, but they are still more similar than to the other bottles of wine. This is also visualized nicely in a PCA, where the 2 bottles of Sauvignon Blanc are tighter, more close to one another than that of any of the other bottles. Similarly, the Chardonnays are closer to one another, and then these 2 blended bottles are actually somewhere in the middle. So we can show that the assay works on red wines, white wines. I also mentioned that I wanted to look into contamination in your wine. And the first thing that came to mind is pesticides. Okay? Pesticides are commonly sprayed onto vineyard grapes to keep, obviously, pesticides away, soil onto the plant itself. Okay? They're all over the place. And there's been a number of publications that highlight how many of these pesticides make it through to the bottle of wine. There's a problem with this. Most of them use very complicated analysis. Either they require long chromatography, 2 hours or more per run, or they require you to have special instrumentation standards, and it can be very, very difficult to do this. I thought, what if I can do this with the PSA in a rapid and quick manner? But to be fair, I needed to look at what was found in untreated wine. So when I went into the literature and dug out a whole bunch of pesticides known to be in the wine, and I searched them in my data set, just based on masses, I came up with a list of 17 that were in your wine in an untreated analysis, meaning that I didn't treat it with the PSA. I analyzed those samples directly by LCMS. If I instead treat them with the PSA first, before doing the LCMS, we get a different set of these pesticides coming out that were unique, that appeared to be pulled out of low concentrations in the wine, and now we can see them. These untreated ones that didn't show up, I can chalk that up to the idea that they didn't bind to my protein. They didn't have strong binding to my protein. I'm more interested in those that did bind that we didn't see before. If you look at the chromatogram for these, I've pulled out and highlighted the structures of a number of them across the board, just so you can get an idea of kind of what they look for. Now, this is based just on mass match. Okay, this is not typically how we do mass spectrometry. If we want to identify something, we like to work with authentic standards so that we can then do fragmentation on the molecules and see if those fragmentation patterns match between the molecule in our sample and the authentic standard. But I didn't have authentic standards. These were pulled from a paper where all I had was mass. However, there is one way that I can look at these. Most pesticides actually end up having either a chlorine or a bromine in their structure. So I'm highlighting one here, myclobutanil, which has a chlorine in its structure. Chlorine and bromines are fantastic in mass spectrometry because of their unique isotopic distribution. Okay? In nature, chlorine has mostly chlorine 35, which is indicated in this peak right here. We call this the molecular ion. And 2 mass Daltons shifted over is chlorine 37, which has about a 30% abundance naturally in the environment. And mass spectrometers are great at pulling apart isotopes. So here you can see that chlorine 37 represented 2 mass units shifted from the original or the molecular ion. So this was a pretty good indication that I had myclobutanol in my sample just based on accurate mass and the chlorine isotopic distribution. But I went ahead and did what we call predicted isotopic distribution calculation, which you just input a molecular formula for myclobutanol, and it will tell you what the predicted spectrum will look like. I show that here in red. And you can see that it matches almost perfectly on the blue lines, which was my actual spectrum from the data. So this confirms with the most certainty that I can have without actually having myclobutanol on my benchtop that we have this pesticide in your wine. And again, this is something that we can pull out with the PSA. With further testing, I can go in and actually obtain authentic standards for pesticides. And in fact, I've done that and started to investigate the data to try and confirm some of these identities. Okay, wine was great to work with, but personally, I'm a whiskey guy. So I thought we should analyze some whiskey. And there's actually a couple reasons again for doing this. First and foremost, in order to produce whiskey, you need to create what's called a mash, or a grain mash, which you do by malting grains, barley, corn, rye, and wheat. And Montana produces the most barley in the United States. So naturally from that, there's been a lot of craft distilleries that have popped up here in the state. Currently there's over a dozen that I know of throughout the state of Montana. But whiskey in itself is actually kind of a bad business model because in order to make the really good whiskey, the 12-year aged whiskey, it takes 12 years. You have to sit on that product for 12 years before you make a dime. Just a bad business model. So what a lot of distilleries have started to do is try and either rapid age, speed age their whiskey, or they try and mimic the aging by rapidly producing or placing the whiskey into barrels every couple of months, new barrels, to try and increase the diffusion of molecules to get that flavor. And so that's why I wanted to kind of delve into it is, could we maybe apply the PSA to give a metric to this speed aging of the whiskey? And furthermore, if you're going to just age a whiskey for a year, because they taste plenty good, maybe I can apply a metric so that they get a consistent product without needing to go to that master distiller and have them taste it. Because what say he has a cold on a bad day? That whiskey might taste terrible, and I don't want to drink that. So let's apply a metric to this. I've had some really good partners on this project, and this is a highlight of one of them. Working with Wild Ride Distillery and 406 Brewing here in Bozeman, we were able to obtain a Montana Growth Through Agricultural grant to start the Montana Research Fermentation Center, or Montana Fermentation Research Center. And through their connections, they got us in touch with Willie's Distillery down in Ennis, Montana. They were gracious enough at Willie's to give us some samples from 2 different experiments that they were kind of conducting. The first was the aging process of whiskey. So they had barrels of whiskey that had aged for up to 9 months, and they allowed us to take some samples from them. When I treated them with the PSA and then did a principal component analysis, I got something that looks like this, where we can actually see that we are seeing an increase in age in the actual principal component analysis, where the squares here were actually freshly distilled the day before we got there. It was a clear liquid. I tried it. It was terrible tasting. The ones over here were 9 months into their aging. They were just about ready for bottling, and they tasted very smooth and very good. So we can actually apply now a metric to their aging process. So over time, we can watch as that whiskey becomes more like the finished product. And again, this would potentially add to the master distiller who's just basing it on taste. Another experiment that they allowed us to take part in was, again, getting back to this kind of speed aging or trying to rapidly get to a product or just changing the flavor profile in general of the whiskey. They had a bottle or a barrel of whiskey, which I'm labeling here as stock barrel, that had aged for about 2 years. They took that barrel and they placed some of that whiskey into a new barrel made by Black Swan, and this is their process. They placed another portion of that barrel and into a new, or another portion of the stock into a new barrel by barrel mill. Excuse me, confusing. They then let some of it just sit in this reservoir tank, and they let it age for another 6 months. Okay, so these were all aged for the same amount of time, and what they were hoping to see is a difference between the stock barrel and the two new barrels, which we can see here on this PCA. And they were also hoping to understand which barrel company would be better to go for their kind of speed aging or flavor enhancement. And you can't really see it in this plot that this barrel mill bottle and this Black Swan are actually very similar. But you can clearly see there's a difference between the stock barrel. So if I analyze these instead by hierarchical clustering, again, where the vertical lines indicate dissimilarity, you can see that the stock barrel is very, very different from the original barrels. But these two new barrels are actually very, very similar. The conclusion we came to is that it wouldn't honestly matter which barrel they placed it into. It would come down to which would they rather spend money in. Did the master distiller have a preference in flavor profile, something like that. Okay? So it was really fun to work in the alcoholic beverage industry for a while, but I really wanted to get back to biology, okay? And I thought, you know, there's got to be something in the lab that we can test that's biologically relevant. And as it turns out, there is. So our lab focuses a lot of its efforts in proteomics and metabolomics, at least in the past, in organism called Cephalobus sulfatericus. It's an archaeal organism from the hot springs in Yellowstone, or the strain that we're working with. And our group has done proteomic and metabolomic analysis when it's under heavy metal stress, viral infection, and oxidative stress by hydrogen peroxide, H2O2. And I thought, you know, this would be a perfect test case for me to try the PSA and compare those results to the untreated analysis that we've been doing for years to see if I can provide either new insights or maybe rapidly analyze the samples in comparison. And that's exactly what I did. So we have a nice little time course that we can lay out where we stress the cells and collect cell pellets over time, and we can watch the recovery at the transcriptome, proteome, and metabolome of these cells. We've done it many times in the past. I took those same samples and analyzed them first with the PSA, then by LC-MS. And what I saw is what you're seeing here. So on the top, I'm looking at the untreated LC-MS. So just taking the cell extract, the metabolites that I could pull from those cells, dumping them on the liquid chromatography, and then into the mass spectrometer. And you'll see here that the black and red trace are biological replicates, two sets of cells grown in different flasks side by side at the same time point. They're very, very similar to one another. Most of the molecules are eluting early on in this liquid chromatography run. Based on the type of LC I'm doing, which is reverse phase, I can say that these are probably fairly hydrophilic. They are not water-liking. Okay? Now, after PSA treatment of those exact same two samples of metabolite extracts down here in the bottom, we can see that there's a bit of a profile shift to things that are slightly more hydrophobic, just based on the type of chromatography that I'm doing. I wanted to make sure that I kept as many things the same in this experiment, and I'm highlighting that with this dashed line of gray. Okay? This is my elution gradient. This is how I'm pulling things off the column so they can be analyzed by LC-MS. So you can see here that as we increase the amount of solvent that's pulling molecules off the column, we're seeing, again, less things in here and more things after PSA treatment. Finally, in keeping everything the same, I did change one thing. You'll notice down here the time scale for this run. 30 minutes to analyze one sample. So 60 minutes to analyze those two samples on the top, whereas 15 minutes was all that was required in PSA-treated samples. So I'm reducing the number of things potentially that would be seen by the mass spectrometer. I needed to verify that this was still going to work then, because I've essentially decreased the amount of time that I'm analyzing my sample, but am I decreasing the amount of data that I gain from this? And when you analyze those cells by principal component analysis in the untreated, you end up with what I call a scatter plot. Okay? Again, each of the colored dots that are the same color, blue, blue, and blue, are the same biological replicate, flasks grown side to side at the same time point. And none of those are clustered tightly together. This is an unbiased method, remember, PCA. So I didn't tell it that they were the same samples. Okay? And it couldn't cluster them together, meaning they're very different from one another. Furthermore, the different colored shapes are not separated from one another, which would indicate that the different time points are resolved. Contrast that with PSA treated samples. Okay? So I've gone from 30-minute chromatography to 15-minute chromatography. I've gone from 850 features or molecules to 240. I've decreased the amount of data in these samples, okay, decreased the number of things I see. I've increased the separation of time points from one another, and I've also increased the clustering of similar biological replicates from the same time point. So this is powerful, right? Works on wine and whiskey, great. Biologically relevant things. Untreated, you can't tell them apart. Treated, boom, you get a nice little graph. I decided to investigate this a little bit further and see if I could gain some understanding of how the PSA is actually doing this. How we can go from lots of features to fewer features and get better separation. So I chose to work with the 2 time points that are kind of furthest apart from one another. 0 minutes, right before we gave them a little bit of stress, and 105 minutes after it had been recovering from that stress that we gave it. And in this PCA, I'm just showing those two. The triangles are the 105 minute, and the 0 minute here by the circles. And you can see that they're separated from one another. This is great. Wouldn't expect it to not be separated based on the previous analysis. If we look at what's called the loading plot, if you remember after I reoriented my mock PCA demonstration, this is what it looks like. These are all those little red circles, the little metabolites. Okay? Again, doesn't make a lot of sense. But if you sit and stare at this for long enough like I did, I could pull out a number of things that were important for differentiating the triangles from the circles. And I've highlighted them here by these squares and triangles. If we look at what those are, okay, and I've highlighted in red the 3 that I found the most interesting, and I'll tell you why. In PSA-treated samples, these 3, numbers 1, 2, and 7, were statistically significantly different. What do I mean by that? They had a fold change of greater than 1.5, so there was at least 1.5 more of that small molecule in the 105 minute versus the 30 or versus the 0 minute. And it had a p-value. It was statistically significant because it had a p-value less than 0.05. These parameters are commonly used in metabolomics. Okay? 1.5 fold change, 0.05 p-value. If we look at those exact same metabolites in the untreated data that I showed a couple slides ago, look at those fold changes. 1, 1.01, 1.2. These are not less than 0.05. These are terrible p-values. So in the untreated data, the fold change seen between these, there isn't any. But after PSA treatment, all of a sudden there's a fold change. This tells me something about how the assay works. And I hypothesized that what was happening is that in, for instance, this 229, the reason that there's 9 times more of it in the 105-minute sample is because not that there's more physically in the sample, right? Fold change of 1, there's exactly the same amount in the cells before we treat them. No, instead what I hypothesized is that something else is binding in the 0 minute that is not present. Or some things, maybe it's many things. Remember, bovine serum albumin or serum albumin in general is a protein that loves to bind everything it can, but it has some selectivity. Maybe I'm making use of that selectivity to go from 1 fold change to 9. So I wanted to test that. I did a binding study to test this. And how I did that is I took my normal PSA assay. I bound molecules to it and let them incubate over 5 minutes. Standard stuff. And I analyzed that by LCMS. I also took and let these metabolites incubate with the protein for 3 hours, 8 hours, and 24 hours. With the idea being that if my hypothesis is correct, that we have different kind of affinities for this protein, different on and off rates. I should see that in this study. It's not just a mass of who can get on it quickly, but maybe who can get on it and stick. Okay? And that's what I'm indicating here with these shapes falling off, is metabolites over time will naturally diffuse. If I'm wrong, everything should fall off, and I'll be left with nothing at 24 hours. I analyzed all of this by LCMS, and I took and selected 9 that I thought were representative of this data. And it indeed confirmed my hypothesis. Okay? Up at the top, I'm highlighting here in blue, metabolites which essentially bound to BSA and then stuck. Now I can't say if that because they have a high affinity, but I can say that it does mean that once they're on, they tend to stick. So I'm going to say that's pseudo-high affinity. At least given the sample that I have. There's also some metabolites that after 24 hours, only about 50% of that metabolite was left bound to the protein. These are things that maybe got onto the protein very quickly, but then slowly diffused off. Maybe they didn't have the highest affinities. And finally, I have things that basically didn't stick at all after they bound. They, for some reason, did bind in the first initial 5 minutes, but then they very quickly came off. So again, this kind of confirms the idea that there are different affinities, or at least on and off rates, to the protein, and this could help explain why I'm seeing this kind of fold change in PSA-treated samples, whereby I didn't see it in the untreated. Again, I don't know what it is that's out-competing that particular metabolite I highlighted, but there is potentially something that's doing that. Now, when you look at these 9 molecules, after 8 hours of binding to the protein, again, after they've essentially equilibrated, they don't really change after 24. If you look at them in those 8-hour time points and you compare their intensities with that of untreated data, so data where I just again analyzed it by LCMS, I'll highlight a number of molecules here to make it a little easier. Let's look at 141. In untreated sample, it had an ion intensity of about 60,000 counts, so I saw it 60,000 times. In the PSA treated, I saw it 600,000 times. Okay, so we've increased by a factor of 10. Right? There are examples where I can see it increasing by as much as 100-fold increase in intensity in metabolites after PSA treatment. One way that I chose to look at this that makes a little more sense is if I take and rank them in a percentile and rank fashion, whereby rank of 1 and percentile of 99th means it's the most abundant ion seen in that set of data. If you compare that same 141 before treatment, it's in the 5th percentile. It's basically non-existent as far as the other ions are concerned. There's way more ions that are more intense. After treatment, it's in the 81st percentile. So it's become very, very abundant within this sample. What this is telling me is that not only do I have different on and off rates, but in general, I'm increasing the concentration of these molecules by, like I said, 100-fold in intensity. So now I have an idea of how this assay is working. It's still a competition for things to bind to the protein, but once they do, I get things that are meaningful. Remember, these things that are more intense in the sample are potentially the things that are important. They're the things that I want to actually know about the biology, to know that sample 0 is not going through oxidative stress, while sample 1 is going through 105 minutes of recovery of stress. Okay, so I'm pulling out meaningful biology using this protein sensor assay. And again, this was with a kind of the best case system. Okay, a system where I have biological replicates that should be very, very similar. In the untreated data, that kind of was a misnomer, but at least after treatment with the PSA, they were exactly what I would expect. I wanted to test this on something that was maybe a little bit more difficult to understand. And in our group, postdoc was working on at the time a model system for hemorrhagic shock. I thought, well, this would be a nice little attempt to try and use the PSA. And I'll explain exactly why I thought it would be good in just a minute. First off, what is hemorrhagic shock? Well, it's your body's overreaction to the rapid loss of blood. What do I mean by that? If you lose a lot of blood, you're not going to go into shock necessarily. It's when your body tries to overcompensate for that rapid loss of blood that you can go into shock. And this becomes important because, one, we don't have any markers to predict if you're going to enter shock. Again, you can't just use rapid loss of blood. That's not good enough. And the bigger problem is that the way we treat shock right now is you just transfuse blood and clotting factors and try and stem the bleeding. But imagine if you're a person who was going to recover on their own without any help. You didn't need transfused blood. In your bloodstream are markers that are telling your body, hey, something is wrong. We are definitely not A-OK, but I think we can recover. Those markers are telling your body that. If you transfuse in normal, healthy blood that has none of those markers into a person that is going to enter shock or may not enter shock, doesn't matter. You're basically diluting that signal that was initially in the bloodstream with happy, healthy signal that tells the body, hey, nothing's wrong. So all of a sudden you have a person that was going to enter shock and you've just screwed up that signaling within their blood. So it's very important to know if they're going to enter shock or not, because you can cause more damage just by transfusing blood. So we want a fast and reliable test to predict shock. Now, I want to put this another way. Imagine Jack and John get into a car accident. Okay? They're both bleeding profusely. They get rushed to the emergency room. The doctor's not going to know what's going on within these two, and he'll probably treat them as if they're in shock. But again, he may actually do more harm than good. So that's where this project kind of comes in. Working with the University of Minnesota, they have a model system for using porcine or pig animals in which they can induce shock into a series of animals, and they can also have a set of control animals. Animals which undergo surgeries which would allow them to then be put through this shock-induced stress, but they aren't. They're just left alone, left to kind of recover. We induce shock in the shock animals by draining a third of that blood and then applying a puncture trauma to the sternum, okay? And then they collected urine samples over 48 hours. Now, the reason I thought this was going to be a challenge for the PSA, and I think we can show that it actually is a benefit, is that urine output across one animal, imagine yourself throughout the day, the amount of urine that you output is going to be different each time you use the restroom, based on how much fluid you've drinking or not drinking, based on how much salt intake you might have. There's a number of factors that increase in one animal the amount of urine output. Therefore, the concentration of metabolites, small molecules in that urine in one person throughout a day varies. We're doing 2 days. That's a lot of variation in one animal. Furthermore, I don't have this nice biological replicate that is going to be consistent. So 2 people at the same time, if they go to the bathroom, are not going to have the same amount of urine. The concentrations are going to be all over the place. And this was indeed what we saw when our postdoc Monica analyzed the untreated samples. I'm showing here 2 urine samples at the exact same time for 2 animals. In this triangle, I'm indicating in the black trace is higher in intensity than the red trace. However, at the star, the red trace is higher than the black trace. So we're seeing this biological variation coming into play. It's not like the Cephalobus cells where everything looked the same before and after treatment. In this case, after treatment, what we end up with is this nice, almost normalized, almost identical chromatogram. The red trace is the exact same urine sample up here, as is the black trace, identical to this. We're seeing an almost identical trace, spare for a little bit higher in the red. I'll take credit for that. I over-pipetted a little bit of volume, okay? We have a slight increase in concentration. But overall, we're seeing almost a normalized set of data. But let's look at it again. I can look at these all day, and I don't know if I can make sense of it. If we analyze by principal components analysis, first off, the untreated samples. Again, a scatter plot, just like we saw with the Cephalobus. We're seeing that there's no tight clustering of individual shapes, like triangles here. They're quite far. And we're not seeing separation of the white control samples and the red shock samples. In this case, we would say that Jack and John, it's going to be luck if the doctors can keep them alive. I don't really like luck, okay? I don't want to just be a guess. I want to have something concrete. And I think after PSA treatment, I can say that that's possible. So again, these are the exact same urine samples that were just previously shown, but first pre-treated with the PSA. You can see that not only do we have good separation of the individual time points, the shapes, but shapes of the same color and shape are clustered tight together. We've done this again by reducing complexity. So in the untreated sample, that was 3,000 features that we saw. Here we're seeing 300. So once again, decreasing the number of things seen, but increasing the important things that are needed for differentiation. Okay? The other important thing here is that at 0 minutes, which are these squares, the animals are exactly the same. Remember, shock hasn't been induced on the red squares, so they should be exactly like the untreated samples. And again, at 48 hours, those pigs have become very similar to one another. The ones that recovered from shock are becoming much like the control animals. But at 45 minutes, 2 hours, and 8 hours, we're seeing a divergence of the shock animals from the control animals. So this is saying that we can actually pull apart maybe something that's relevant to us being able to fix the problem. I decided to investigate the 2-hour time point further to see if I couldn't find a marker of potentially stress in these animals, or excuse me, shock. I chose 2 hours because I found it to be medically relevant. If you do get in that car accident, it's going to take time for the ambulances to show up, ambulances to transport you to the hospital, doctors to run a quick test, get the analysis. I figured 2 hours was an appropriate amount of time for this to take place. If I analyze the 0-minute time points versus the 2-hour time points for both control and shock animals, I still see separation of those animals. Again, the 0 minutes are clustered right on top of each other. Here's our shock animals at 2 hours. Here's our control. So we're seeing separation. Now, these animals, these 3 animals that I'm showing for control and 3 animals for shock, were run at the same time of the year. Okay? So I thought, well, maybe I'm just getting lucky. Maybe these samples, for some reason, are just going to be similar to one another. Well, luckily, Monica and the group in Minnesota had conducted a second experiment a year later with the same type of setup, shocking animals, watching them over the course. So I got another sample of urine from 2 hours post-shock, and I did a blind test. As indicated by this black dot, it clusters right where it should with the other shock animals. Remember, a year later. So this means that I'm actually getting a viable test that could be potentially used across, tens of years we're talking about, and we could get the same clustering. Again, I wanted to investigate, well, what's doing this separation that we see up here? So if we look back at that loading plot, of all the features, we can see which ones are kind of important. I've numbered them 1 through 15, excuse me. I searched these features, again, they're just masses. I searched them using a database called the Metlin. And what I got out was this list, and you can see that there's a lot of formulas here, or maybe just a generic peptide formula. This is not super informative. It doesn't mean I can narrow it down to a compound, which I can then go buy an authentic standard for to validate. It's kind of just, okay, now I got to start, kind of going through this data. But there was one that was really interesting. Number 1, creatinine. Now, you can see here that it's definitely important for differentiating the 0 minute from the 2 hours because of its placement down here. But why is creatinine showing up as something that's important? Creatinine is something that should be in your urine. It's the degradation product of creatine. It gets excreted through the urine and it goes away. In fact, creatinine is how normally you normalize data sets. 2 people that give urine, they look at how much creatinine you have and normalize everything else to that creatinine level. So why am I seeing creatinine as an important potential marker of shock? Well, I thought it got back to this idea that potentially creatinine's binding and then something else is competing for creatinine in the other set of data. So maybe it gets back to the same thing I saw with Cephalobus. Before I get ahead of myself, I really do need to confirm that we have creatinine in the sample. So I bought an authentic standard and I analyzed it by LCMS. The gray trace shows the creatinine standard run by LCMS, the total ion count, and the black trace shows the putatively identified creatinine in my urine sample. Well, that matches pretty good, but we like to take it 1 step further as mass spectrometrists and do something called fragmentation. So what we do is we take our creatinine and we hit it with energy, or we hit it with a gas that's been ramped up with energy. We like to do this at 2 different voltages of energy because we want to see how it fragments with different amount of energy given to it. So at 10 volts and 20 volts, the fragmentation pattern, the way that this molecule falls apart given enough energy in the urine sample and in the creatinine standard, and in fact in the urine sample spiked with creatinine, looks exactly the same. 10 volts, 20 volts. So this confirms that we have creatinine. Now, this is not something novel, right? Creatinine is supposed to be there. I was just showing that it truly was this thing. But then it gets back to this idea. Why is creatinine appearing to be some sort of marker for shock? Okay, there's much more creatinine in the control samples than there is in the shock. I think it's something like 4-fold more creatinine in control animals. Well, again, I got down to hypothesizing, which may or may not have been a good idea. And I came up with this idea, and I remember I brought it to Brian, and I think we nailed it. I mean, we sat down and we hashed this out, and we've looked at this data for a very long time. And what we came up with is, in the control animals, creatinine binds to BSA. Okay? Doesn't matter how much you have. In fact, if you look at the untreated samples at 2 hours, creatinine levels are all over the place. There is no pattern for creatinine in the control samples, or in the untreated. In the control samples after PSA treatment, we see creatinine binding. I'm overrepresenting the amount that's probably bound. But something or some things are coming along and kicking that creatinine out of that spot. Or maybe they jump onto the protein faster. Maybe they don't diffuse out as quick as creatinine. Again, it kind of gets back to what I was showing with the Cephalobus data, that we have a couple of observations that would point to this being true. And what I think is happening is these markers of shock are still out there, and I need to go find them. And again, it might be multiple things that are out competing creatinine. But I came up with an idea for a potential clinical test. Again, we're trying to find something that would be useful in the medically relevant fields. There's something out there known as an enzyme-linked immunosorbent assay, or ELISA. And these are very, very good assays. They get used all the time for the reason that you have an antibody which is going to be specific for your molecule of interest. And they make these so that they have high specificity. There's a very low false discovery rate, okay, or a negative result in this case for a patient's test. The way that they work is you have an antibody that's bound to some sort of well, I'm just showing it here kind of in like a beaker. You have your marker or markers of stress, whatever it may be, coming in, and they bind to this antibody. A secondary antibody then comes and caps this and allows for this red antibody, which has an enzyme linked to it. And this enzyme turns over a colorimetric substrate. So basically, only when the triangles are present and bound to this right here, and then you get this event and this event, you get a color change. Makes it very easy for doctors to say yes or no, a patient's going to enter shock. No, a patient's not going to enter shock. They have very good statistics as well. And it takes about 10 minutes to do one of these assays. Okay? This would be a rapid test, exactly what we were looking for. So moving forward, this is something that I'd like to explore. All right. With that, I'd just kind of like to recap some of the reasons why I think this PSA is really important and why you should care. When you're looking at, sorry, I lost Valerie. There we go. When you're looking at untreated samples of Cephalobus going through oxidative stress, I showed this. We get very clear separation of time points and clustering. We get great data. The same can be said when we have shock animals. Okay, I showed that as well. You don't really get any sort of nice clustering in untreated samples. One thing I didn't actually show is the wine data before and after treatment. In the wine data, it's a little different. So if you remember back to my first set of wine data with just the 3 different varietals of grape, we can see here that in the untreated sample, we still get separation of the colors. It's the shapes that become blurred. We lose that temporal resolution, which is something that we clearly already have here. And again, in this wine data set, we've gained that temporal resolution as well. So with that, just a couple of quick summary points before I finish. With the Cephalobus data and the shock data, I was able to show that we can simplify biological systems, specifically complex biological systems. We can decrease the amount of data seen here by as much as 10 or maybe 100-fold in some cases. It's fast and reproducible. What I mean by that is you saw the traces for both the shock and the Cephalobus data. They were very reproducible. And the chromatography was upwards of 2 or 3 times faster than in the untreated samples. It concentrates samples. Remember I said I increased certain metabolites by upwards of 100-fold. I've concentrated them. And I think we can see that it can be adapted for a high-throughput screening method. Finally, what we're actually getting is true markers, true fingerprints, true profiles that can differentiate 2 sets of data from one another. Okay? So I like to think of the PSA as a sponge, a selective sponge. Soaks up molecules of interest and then spits them out for me to find. I'd like to give a couple of acknowledgments. The Bothner Lab, specifically Brian, he's been a great mentor. You know, he gave a great intro to this defense, but really it's been awesome to work with him because I've come to him with lots of crazy ideas, and provided it wasn't too crazy, he let me run with it. It's why I'm into things like 3D printing, and it's why I'm into things like this assay. Is we got to explore all kinds of avenues, and I've gotten to do it all because he's been so supportive. Monica, again's our postdoc that was working on the hemorrhagic shock data, and she allowed me to use some of her samples. Josh was basically the guy who trained me on everything. Trained me how to use the LCMS, trained me how to do metabolomics data analysis, so I owe him a great deal. 2 undergraduates specifically have worked on this project, Courtney and Catherine, and they've done some fantastic work. I want to credit them for that. The rest of the Bothner Lab is always there supporting me. I've had other projects and graduate students and undergraduates that have helped me with those projects as well. My graduate committee, Valerie, Ed, Michelle, and David, thank you for being there along the way, reading through my dissertation. I really appreciate it. The mass spec facility, Jonathan and Jesse, Basically, they are the reason that I have this data. They keep the instruments running for me, and that's something to be said considering I oftentimes end up breaking them. In my third year, I was awarded a Capriva Fellowship that allowed me to travel to a conference specifically on the wine pesticide. I got a whole bunch of cool ideas. I got in contact with a number of people that provided authentic standards for me for this project that I can now pursue. So I also was able to get a really nice computer that could analyze these data sets. Again, it's very complex data. So I'd like to thank the Capriva family and College of Letters and Science, as well as the selection committee for awarding me that grant. In the liquor side of things, Phil Sullivan over at Wild Rye Distillery, he got me in contact with a number of people, including Willie's Distillery, and then Doug over at the Wine Gallery for providing not only the wine, but also developing the selections for us to test. That's been really, really great, rather than just me going into the liquor store and buying whatever I felt like. And finally, family and friends being supportive, helping me along the way, whether it be research related or just kind of being a friend. So with that, I'd like to thank everyone for your attention, and I'd be happy to answer any questions.
Well, thank you, Tim. And I realize I was remiss in my introduction on a couple of points. One, I'd like to mention that Tim's parents are here, Mike and Lori. They were able to make the trip out to see the defense. And I'm glad you brought up the Capriva family, because this really was a unique case where we couldn't do a lot of this work off of normal funding. So we needed some external funding, and so we were able to get this from a couple of different sources. And so there's a patent pending on this process that the university decided they were not interested in pursuing after the preliminary filing. And so what does Tim do? Tim says, I can write a patent. And so he largely wrote the patent. I helped out, but it was really his writing of the patent too. So if science doesn't work, I become a patent.
Patent attorney. It's my backup.
Yeah. So, and he makes a really mean Nutter Butter peanut butter cookie. So, question please.
Jonathan.
So the fragmentation patterns on your drafting standard look to be really useful to confirm that was the molecule Douglas. How many fragmentation patterns are already known or in databases for your pesticides for the wine project?
Yeah, so that's a great thing about a database called the Metlin database. It's produced out of Scripps, and a lot of their data comes from either pharmaceutical companies or pesticide, either production companies or people looking to test pesticides. So there's a number of them that are in that database. Unfortunately, none of them match the original data set that I was looking at that were of interest in the PSA-treated samples. And so what I've actually been able to do is get a hold of the agricultural department over here, there, the corner of Grant and 11th, and they provided me with a sample of about 100 pesticides that I can actually fragment for real data. Andy?
In the beginning of your talk, you showed sort of a predicted mass spec of one of the pesticides that you were looking for.
Mm-hmm.
And it seemed like the predicted one overestimated pretty consistently by about 1/10 of an atomic weight unit. Can you?
No,
Oh.
I shifted it so it was easier to see.
Oh, okay.
Yeah, the actual output was spot on, but
Okay.
when you're looking at it, it can be a little bit hard if they're smack dab. So I pulled them apart.
So how are those simulations done? Is that just basically an analysis of how much energy you give the system and the individual bond strengths, or?
No, that wasn't a predicted mass MSMS. That was just a mass spectrum. So that just looks at a charged species.
Okay.
So it's just based on calculating. It's a calculation of the first off. So like the molecular ion, the very strongest ion that was present, is using the most abundant elements in each case. So chlorine 12, nitrogen 14, oxygen 16. And there's some decimals after those ones. And again, chlorine 35. Sorry, carbon 12. And then what it can do is it multiplies the amount of carbons or oxygens or nitrogens, hydrogens seen by the other isotopes that are known to exist in nature, which are far fewer, except in the case of chlorine.
Okay.
Yep.
So I would think that there'd be a real advantage if you are trying to do fragmentation in simplifying the pattern, because when you have these complex mixes, there's not enough time to do fragmentation.
Right.
Did you explore that at all and seeing how much benefit there is to the PSA in getting fragmentation of interesting compounds?
Huh. No, we. And especially because you enhance the concentration of them. One of the biggest problems is not only you have a lot of overlap with other things, but you have very little of it, and you have very little time to fragment it before it comes off the column, unless you're doing direct infusion.
So I think you should add that to your test.
I think that's a great idea.
Yeah.
We should definitely add that.
Yeah, because I think it's very powerful to be able to pull out just a few things and have a much better chance of fragmentation.
Yeah. That's a really good idea, actually.
We'll run and fix that right away.
Yes, we will. Monica.
Do you use PCA assay to run the biological matrix
So we've attempted, and I didn't show that data. We've attempted it in blood, and we're getting. I think it'll work. It requires more effort on my part in actually prepping those samples. What we were seeing is, blood contains, whole blood contains cells which you can spin out, but then it still contains a large amount of protein. And so you have to go through a second process to get rid of that protein. And you're essentially eluting things off of that. But then you're still left with a lot of salt. And I'm not sure how much of a role that's playing in the effects on this. I haven't had a big enough data set. We're working with Ed, and we're trying to get a hold of a good data set to try this on, but in the past we haven't had the best success. I think I can get it to work. What's that?
Gary promises coming next week.
Awesome. So next week I'll let you know.
So, I have another question. This is a hard one, because I've had a lot of people explain to me what a PCA loading plot is.
Yeah.
And I've never really been able to grasp it, I confess. So can you take a crack at explaining what a loading plot is?
Sure. So I'm going to go back. I'm going to go back to my little diagram because I think that's simpler. Oh, that's not what I wanted to do. Okay. So again, if we have a simple data set, just 1 and 2 axes, okay, where along this axis is the number of counts of metabolites in sample 1, this is the number of counts of a particular metabolite in sample 2. And we do this reorientation whereby we take and put the most variation along this axis, right? That makes sense. We're trying to find the place where we can find the biggest variation between any 2 data points. And we do the same thing along this axis, okay, so we create a 2-dimensional type of plot. We've oriented it into this eigenspace, okay? Now, these are still the raw data points, and what I should mention is that in order to take it to the next step where we put it into the loading plot, is we score each one of these for how much they contribute to, for instance, the first principal component. So we could assign this a score of 10 for the first principal component, this minus 10, and this would be like a 0. It contributes nothing to that component. Then what we do is we have now a ranked system for which metabolites are the most important for contributing to these data sets. And that is in essence what your loading plot is. To take it the next final step, to go to individual data points, what you do is, I've highlighted these, but you can do it with, you do it with every single data point. You go back to your raw data. Let's say we gave a score of 10 to this data point, and in the sample 2, which is the blue side, let's say it had an intensity of 10. So we multiply the score by the intensity, and we get 10. Let's say for this one, we gave it a score of 8, but the intensity was 1. So now, this would get a score overall of 8. We do that for each data point in each sample along the first principal component. We do the same thing, scale all the things in principal component 2, and you sum up all of those numbers. 8 plus 10 plus 12 minus 7 minus 2. Sum all of that up, and that's what gives you this final data point shown here. Is a new coordinate system based on the summation in component 1 and the summation in component 2. So in this case, the summation of all things scored and scaled would be something like 10 minus 1. Does that make sense? So the loading plot can be thought of as all of the metabolites, but they've been scored.
Better.
It's a complex analysis. It is. Yep.
Back just to hammering up PCA more, what are the actual units of those axes? Do they keep the units of essentially the 2D plot, or do they,
Nope.
do they make them arbitrary? Or what?
Arbitrary. It's in eigenspace. So we've generated eigenvectors. And so it's a unitless space. We simply call it the amount of variation that is consumed by the data in principal component 1. So if you look on any one of my plots, I show that here. So always the first principal component has the most variation among it. So this contributes to almost 80% of the variation along that one component. And then you can see that number decreases as you go down the different components.
So then what's to stop you from just changing the arbitrary scales of your axes to make things look farther apart or closer? I'm not trying to accuse you of anything, I promise. I'm just curious.
Well, so that's one of the downfalls to this is ideally if I had this plot, and let me show you a great demonstration of that. This will be faster. In that whiskey data, the barrel data, in fact. Because here, look at 60% of the variation is here and 20% is here. So ideally, these are scaled in a box, right? But the way that it should actually look is a compressed rectangle. You can think of this percentage as a new axis. So it could be 60, units long if you want to think of it that way. And this would be 20 units high, so you get a box. So it actually skews it. And that's why this is misleading, whereas the clustering shows you really what's going on. Because to me, in this picture, the triangles and the circles look like they're about as far from one another as they are from the squares. But that's not actually true.
So then do I read that as saying that the triangles are 60% dissimilar than the squares? Or
I don't know if I'd apply a percentage necessarily directly to this. It accounts for 60% of the variability of all metabolites, which could be contributing from the circles as well.
okay.
But if you want to think of it that way, it's not, I don't want to say it's wrong, but it's not,
Not wrong, but it's not right.
yeah, exactly.
Okay, thanks.
Okay, I think with that, we should thank Tim one more time. Yeah, we need some time and some questions for the professor.